Hey guys, Mr. P. In this video we're going to talk about DNA condensation and movement of chromosomes, specifically focusing on the IB understanding of D216, which states DNA condensation and movement of chromosomes as a shared feature of both mitosis and meiosis. This understanding includes the role of histones in the condensation of DNA by supercoiling and the use of microtubules and microtubule motor proteins to move chromosomes. So the first thing we need to talk about when talking about how DNA condenses is basically talk about how DNA is structured. We know that at the base level, DNA consists of a long chain of nucleotides. Those nucleotides are going to be oriented into a double helix structure, which is a structure that is twisted. It is double-stranded, meaning one strand is running anti-parallel to the other. This DNA structure, anti-parallel double helix, is going to be wrapped around histone proteins. These little purple dots are called histones. Those are proteins that are going to assemble into an octamer, meaning they're going to assemble into a group of 8, which creates a nucleosome. Once the DNA, which is that double helix, is going to wrap around that octamer of histones twice. So condensation involves DNA being wrapped around histone proteins. The DNA is going to wrap around the octamer twice. It's going to produce the nucleosomes. That is going to become the first quote-unquote supercoil by stacking all of those nucleosomes on top of each other to form this long strand of supercoiled DNA known as chromatin. The nucleosomes form as a result of the grouping of those 8 histone proteins and wrapping the DNA around it twice. Each of these little groups would be a nucleosome. Nucleosomes are linked by a section of DNA that isn't wound called linker section or linker DNA. Once all of those nucleosomes are wrapped tightly around to produce this 30 nanometer helical fiber and supercoiled again to form the 300 nanometer fiber, it is going to form that familiar chromosome structure that we see during times of active cell division, specifically mitosis and meiosis. This condensation structure ensures that different DNA molecules can be transported in one package rather than being spread out all over the nucleus. It is really important to note, and we've said this in past videos, that chromatin, this state here, is going to be the inactive form of DNA, meaning when cells are in interphase and they are inactive in terms of their cell division, they're going to have a lot of DNA kind of spread out all over the nucleus. But in times of active cell division, when the DNA needs to be activated, it's going to supercoil and tightly wind into this chromosome structure in order to package a lot of DNA in a really small amount of space. It also makes the DNA a lot more manageable in terms of moving that particular segment of DNA around and ultimately dividing the cell. So, how is the chromosome going to be moved once it has been condensed? The movement of chromosomes is dependent on the ability of motor proteins to join or connect to these microtubules and ultimately move them by sliding them past each other. So specialized molecules called motor proteins push or pull objects around a cell. These structures right here are the motor proteins. They consist of protein and utilize ATP in order to dissociate and move their conformation shape in order to produce cell movement. They use microtubules, which are these kind of green and white structures, as tracks, or they can attach 2 of these microtubules like they're doing in this particular image to get one to slide past each other. Obviously, this arrow indicates that this microtubule is going to be moving this way, whereas this microtubule is going to be moving this way. They can elongate the cell or they can shorten the cell based on causing these microtubules to slide past each other. Motor proteins use ATP, which is adenosine triphosphate, in order to produce a conformational change, which means they are going to cause a shape change that moves the microtubules. These motor proteins can actually cause a breakdown of ATP into ADP and phosphate, which causes these arms to disassociate or let go of the particular section of microtubule that they are attached to and grab the next section in order to shift this entire thing in one direction. Okay, and so if we focus on how those spindle fibers and motor proteins ultimately move chromosomes and ultimately move cell, we have to kind of focus on a variety of different microtubules. All of these green structures are microtubules. There happens to be 3 different types. Astral microtubules are going to be the microtubules that reach out from the centrosome. These structures are called centrioles, and when the centriole produces these spindle fibers, which are the microtubules, it produces the centrosome. And so the astral microtubules are the microtubules that are reaching out from that centrosome. The next kind of microtubule would be the kinetochore microtubules. The kinetochore microtubules are a lot longer than the astral, and those are the microtubules that will bind to the kinetochore, which is a small protein that is on the surface of the centromere. The centromere is the protein structure that binds the 2 sister chromatids together. Now, on the centromere is a binding protein known as the kinetochore. And so the kinetochore microtubules are going to be microtubules that connect to that kinetochore and therefore allows the centrosome to bind to the chromosomes in order to move them about the cell. The third one would be the polar microtubules. These are the microtubules that do not connect to the chromosomes but rather pass beside them. So you will see that on this diagram there are kinetochore microtubules, which are the microtubules that connect to the chromosomes. You can see them here, you can see them here, here, and here, but you will also notice that there are polar microtubules that skirt in between the chromosomes and connect one centrosome to the other centrosome. These are the microtubules that will be associated with the little motor proteins that allows the poles of these cells to be pushed out away from each other and allows the cell to elongate. It also is going to allow these sister chromatids to be separated because they are also connected to different microtubules that will be shortening at the same time. So if we focus on all of the movement within a cell and we take out all of the unnecessary stuff and we focus on just 2 individual centrosomes, you will notice that there is centrosome in the left pole. There is also a centrosome on the right pole. You will notice that we have some of those polar microtubules that are going to go between the chromosomes. We also have some of those kinetochore microtubules. The kinetochore would be these little green structures which are hooked to the centromere, which is holding the 2 sister chromatids together into the shape of DNA known as the chromosome. That is the active form of DNA, and it is active during times of active cell division. So what happens is these kinetochore microtubules will actually shorten, meaning they're going to be pulled towards the centrosome and will split the sister chromatids apart because they are being pulled apart. They're being pulled in opposite directions. These kinetochore microtubules will be shortening, which does nothing for the overall length of the cell, but it pulls individual chromatids towards what will become the 2 new nuclei of the cell. Once we package these 2 chromosomes into a nucleus, we now have divided the nucleus into 2, and we can divide the cell via cytokinesis in order to produce 2 cells from 1. While these kinetochore microtubules are shortening, okay, this one going this way, this one going this way, this one going this way, and this one going this way, we also have some of those polar microtubules which are actually going to be hooked together by these motor proteins. Those motor proteins are going to be utilizing ATP in order to push this microtubule towards the pole and push this microtubule towards the pole while simultaneously pushing this one towards its pole and this one towards its pole. Because these microtubules can slide past each other, it elongates the cell overall, causing the cell to form what will become that cleavage furrow in the middle, which will eventually split into 2 daughter cells from 1 cell and will package these new chromosomes into new nuclei in order to divide 1 cell into 2. Now, in the next video, we're going to go into the intricacies of mitosis as a whole, and we're going to look at the process of mitosis, including the subphases that make it up. But for now, this video needed to talk about why DNA condenses, how DNA condenses, and needed to kind of set the stage for how DNA is separated and how the cell is split before we go into the sub-processes of mitosis. If you learned something, give it a thumbs up. Subscribe to the channel, leave your questions in the comment. We'll see ya.